My talk will also be about some application of opossum pipeline, but in this case it will be applied on population genetics data in order to predict the development of complex diseases in different populations. So in 21st century, one of the most common reasons of death are actually non-communicable or non-infectious diseases. At the first place are cardiovascular diseases, which are followed by different type of cancers. All non-communicable diseases, specifically complex diseases, in their development have genetic component. Most common genetic factors which increase risk of disease development are single substitution in DNA sequence, which called single nucleotide polymorphism, or short SNPs. But however, the genetic factors are not enough to develop complex diseases. There is also a crucial influence from environment and lifestyle. But going back to genetic factors for diseases can be rare mutations, which cause Mendelian diseases. These rare mutations are characterized by very low frequency in the population. However, they have very high effect on disease development, which means if a person have this rare mutation, he will develop also the disease. But other type of genetic factors are polymorphisms, which cause complex diseases. So this polymorphism has very high frequency in the populations. However, the effect size of each polymorphism or SNP uniquely is very low. So identification the set of SNPs which cause and are risk factors for common disease development is a very challenging task. One of the common used approaches up till now is genome-wide association study. This study basically take the people with certain syndrome or certain disease and the control group, and they perform whole genome analysis. It can be a whole genome sequencing or genome-wide microarray analysis. So then identify the pool of SNPs which are significantly have high frequency in the case group rather than in the control. And this set of SNPs are considered as associated with this disease development. However, the genome-wide association studies has also its limitations. Up till now, GWAS have been performed on limited number of populations, basically mainly from Europe and Africa. But here is the problem. When the study is repeated on the other populations, somehow the results are different. So the certain SNPs which are reported as associated with certain disease for one population can have no effect or even be protective for the others. So taking into account this issue, in this study we have decided to describe whole set of disease-associated SNPs in different populations, and then evaluate the contribution of risk alleles in the patients, taking into consideration the population belongingness of that individuals. So for that purpose, we have used publicly available data. The genotype data for people from 51 population were taken from Human Genome Diversity Project. And importantly, in this study, we have used genotype data for Armenians, which is basically understudied in these kind of researches. And then we have filtered from the primary data set and selected only that SNPs which are associated with different complex diseases. And the final data set is the matrix with the, oh, it doesn't work. What did I miss? So the final data set is the matrix with the genotypes. So in order to analyze this matrix in the future, we have encoded all the genotypes into the numbers. So the major alleles, homozygote major alleles were encoded into 0, heterozygote alleles 1, and 2 for homozygotes minor alleles. Thank you. So then we have performed self-organizing maps-based analysis. So Harry has already explained how the pipeline. I will just mention some specificities we used in this study. So input data of our study is the genotype matrix, where the columns are samples from different populations, which are grouped by geographical regions, and the rows are the SNPs. So here we can see that each SNP has unique profile across the samples. And you can notice that some SNPs have similar or even identical profiles. So the sum will reduce the dimensionality of our input data and simultaneously perform the clustering. And as a result, we will have these clustered SNPs on our result map. So in our map, each pixel is the microcluster, where are collected the SNPs with similar profiles, and each pixel is colored according to its value. So the pixels enriched with major allele are colored blue, heterozygote green, and minor alleles are in red. So this is the final map where we can see the red spots, which are the spots for minor alleles. And it was reported the majority of minor alleles are actually the disease-associated alleles. So in future analysis, we are interested only in these red spots. So these kind of portraits are generated for each individual in our data set. Also, the mean portraits are generated for geographical regions, and the summary map, where we can see the overview of all our samples. Here we can see each clusters and perform downstream analysis separately for each cluster. So this is our results. We have here the mean portrait of all the geographical regions. And you can see that the portraits are very similar for the regions which are located geographically near each other. And in the middle we have the minimum spanning tree. This is the tree where each dot is one individual, colored according to its geographical location. And it's based on Pearson correlation between some individual some portraits. So interestingly, this tree is reply the geographical overview of actual location of this population. And in order to study a little bit more deeply Armenian population, we have performed the deep sum analysis only for Armenians, population from Middle East and Europe. And here, an interesting part is that Armenia in the minimum spanning tree is actually located directly in the middle of population from Middle East and population from East Asia, which is the actual location Armenia in the map. So this is the summary map. Here we can see 13 clusters or 13 spots. We can divide these spots into two groups. So the spots which are specific for one population, and the SNPs which are located in that spot are actually characteristic for that geographical region. For example, this region are for Africa, and the other part, other group of the spots which are mixed spots and characteristic for pool of group of population, and they show the similarity between that population in terms of disease predisposition. So then we have performed the downstream analysis for each cluster, this 30 cluster individually, and it's a little bit small, but if you can see, this is the list of diseases which were enriched in each cluster. Here we can distinguish the region-specific diseases and also some common diseases which were common for all the geographical region. Here was immune system cancer, diabetes, and different mood disorders. So then we have deeply investigated the Armenian population and found one specific cluster which are characteristic only for Armenian samples. So this is the mean allele score, distribution of mean allele score across the Armenian neighboring population for the SNP specific for Armenia. And this is its distribution across the world wide, where you can see that Armenian population actually have very high mean allele score for their specific diseases, SNPs. And here is also the list of Armenian specific diseases which were reported during this analysis. So the next step of our analysis were investigation more deeply the diseases. For that, we have selected one type of disease and so we selected immune system cancer because they were common for all geographical regions. And then we identified 700 unique SNPs associated with different immune system cancers and perform additional SOM analysis using only that SNPs. Here is the geographical mean SOM portrait for that SNPs, and also you can see that similar portraits are very similar for neighboring populations and very diverse for far ones. And the global portrait is a little bit replicate the primary global population portraits. But if you are going deeply in each type of disease, you can see the variety of mean allele score in each subtype of immune system cancer type. So in this case, here are the population. Each color represents the geographical region, and here is the mean allelic score for that particular disease. And for example, for Hodgkin lymphoma, you can see that African population have very high mean allelic score compared with all of others. And the vice versa, for example, for multiple myeloma, the population from Europe, Armenia, and Middle East have high score compared with all of others. And also it was interesting to compare the real immune system cancer incidence and the genomic risk score. Here is a map of the incidence worldwide reported in last year. And here is the average genomic risk score for all immune system cancer in all geographical regions. You can see a little bit correlation between incidence and the genomic risk score. However, it's important to mention that in this analysis we have reported only genetic risk, but development of the complex diseases, which are also the immune system diseases, also influence lifestyle, environment, epigenetic factors, which are not considered here. But however, we wanted to try to evaluate the impact of risk alleles in this disease development. So going deeply, we have taken also the case control data and tried to predict their risk. So we have taken the data for 3 types of non-Hodgkin's lymphoma and the corresponding controls. All our samples were from Europe. So here is the background portrait of the geographical regions, which were construct based on immune system cancer SNPs. And here is the predict portrait for the controls and the predict portrait for each type of lymphoma. And here you can see that we have one spot which is very specific for the population because we know that our individuals were from Europe. And if we eliminate this population effect, in all the cases we can see small other spots which are considered really specific risk alleles for that kind of that disease. So in order to eliminate this population effect, we have from each type of lymphoma made minus controls. And here we can see that the European spot, which were population-specific spot, is like less enriched, so they are collected only minor SNPs. And there are some spots with minor alleles, which were not population-specific spots. And interesting fact to mention here that all the other spots which were considered a real risk alleles, risk SNPs, are located in Africa-specific region, in South Asia regions, but our patients were from Europe. The same picture is from other two type of diseases. So in a conclusion, I want to say that we see that there is a region and population-specific pattern of disease development, which is very important to take into account in the disease diagnosis. Also, it is very important to enlarge number of GWAS target population. And for example, for Armenian population, we don't have any information, and at this point, we can't really predict the disease predisposition for Armenian population. But on the other hand, population level genetic risk score is very powerful approach to predict the diseases. And the some portraits can really have its very good contribution in this kind of studies. So I want to thank my group, bioinformatics group, and also people from Interdisciplinary Center of Bioinformatics, and all of you.